Okay. Ooh, it works. You all know I can speak kind of loud, so I have to hold this away. Before I introduce today's speaker, announcements from the audience.
Hey everyone. On Thursday of this week, we have an opportunity to meet some folks who run the ARGO program, which is a program out of WHOI, the Woods Hole Oceanographic Institute, that deploys floats in the global ocean, often off vessels of opportunity like container ships. And these floats send information on ocean currents. There are like hundreds of floats out in the ocean right now, and we can track them. They give us a lot of like really important oceanographic data. And Thursday at 1:30 this week, there will be a chance for us to sign the floats that will be deployed this summer off of the TSOM. So the TSOM is the vessel of opportunity for 6 Argo floats this year. So 1:30 down at the waterfront. I'll send an email as well. If anyone is interested in meeting with the Argo folks ahead of time on Thursday, so before 1:30, just send me an email and I can help to arrange that as well. Okay, thanks.
Anyone else? Ooh, what did I kick? I'm nervous because this is such a thrill for me to introduce our speaker today, Alexa Cacacie. Alexa is a 2020 graduate of Maine Maritime Academy. She got her degree in marine biology. While Lex was here, she was an award-winning middle blocker for the volleyball team. So she had a great contribution there. She did a lot of great internships and other research experiences, which you can ask her about afterwards. After Lex graduated, she went up to Alaska, and she served as a fisheries observer on the fishing vessels in the Gulf of Alaska and the Bering Sea. And that, I suspect, is what gave her a taste of what it's like to be a fish biologist. So, of course, that makes me very happy, too. After she left the observing program, she was I had to write it down. She was a stranding technician for Marine Mammal Stranding Center in New Jersey. So she worked with them for a while. And then in the summer of 2022, she started her master's degree program at the University of New England. And it's that master's thesis work that she successfully defended about a month ago. She'll be graduating this next month with her degree this month. May. No, it's April. Next month with her degree. But that's what she's going to be talking about today, is the research that she did for her master's thesis.
All right. Are we good? Can we hear me? All right. Perfect. Awesome. Well, thank you, Cleveland. Today, first of all, I just want to thank all of you guys for coming. I know some of you have to be here for class, but I hope it's at least worth your while a little bit. So today, like Anne said, I'll talk to you guys a little bit about my master's thesis, quantifying ontogenetic shifts in habitat use in rainbow smelt using otolith geochemistry. And so first off, I kind of just want to acknowledge my funding sources and my advisor, my committee, everybody who I've worked with on this project has been extremely helpful and really wonderful. So especially Sea Grant New Hampshire for even giving me this opportunity. Buttons. Okay, so to start off, we just need to discuss a little bit about life history strategies of fishes. So just like us, fish have different life history strategies to be able to adapt to their environments. And so diadromy is a life history strategy which requires migration in order to complete their life cycle. So the prefix dia in Greek means across, and dromos means running or path. So if we put that word together, diadromous, it means to run across, which is exactly what we see in fish. We see them moving across different habitats. So diadromy is defined as a predictable migration between marine and freshwater environments. And while a lot of management and publicity is focused on really popular diadromous fish like striped bass and all of those fancy recreational fishes that we all know about, less than 1% of all fish species are actually considered diadromous. But how do we actually determine through evolution if a fish becomes diadromous? So first of all, we have to have the ability to migrate in the first place. If we don't, then we don't evolve to be diadromous. But if we can migrate, we have to look at energetic costs. Just because a fish can migrate doesn't mean it should. So if we experience high energetic costs, then again, we don't evolve to be diadromous. But if there are low energetic costs, then we must take fitness into account next. And so if a fish moves into a new environment, does this increase or decrease their fitness? If their fitness is decreased, then diadromy doesn't evolve again. But if there's some sort of increase in fitness, then eventually we get the evolution of diadromy. So anadromy is a type of life history strategy that falls under the much broader category of diadromy. So anadromous fish migrate from saltwater to freshwater to spawn in order to complete their life cycle. And so some local species here in New England that exhibit a kind of necessary migration, otherwise known as obligate anadromy, are the Atlantic salmon, the rainbow smelt, and the Arctic char. So the ability to osmoregulate or regulate the amount of salt in their tissues is essential to anadromous species because salinity experiences are going to vary across different systems wherever the fish is. So there are a lot of benefits to being able to move between different environments, and the ability to form intrapopulation groups is one that has been investigated since about the 1960s. Which brings us next to the idea of the contingent hypothesis. So Clark's contingent hypothesis is the idea that intrapopulation groups form to increase overall fitness within a population. So on this figure here from Secor 1999, we can follow the bold arrow. So I know it's a lot to follow, but so if we follow the bold arrow here, we see a traditional migratory pathway of a population, right? Everybody kind of follows the other. But if we look, we can also see these little groups breaking off onto the side, which is what we would consider to be contingents. So by utilizing contingents, should anything happen to the population and wipe out one portion of the population, the other contingents existing in different environments have the ability to survive. So in dynamic environments such as estuaries, system instabilities promote increased exploration of other habitats within that system. And so for a little bit more of a visual example, here we have larval dispersal of rainbow smelt from spawning habitats to nursery habitats, where they'll eventually grow into juveniles. And then we have migration from nursery habitats to adult habitats, where juveniles will hang out, they'll mature to adults, but we also have some slower growing juveniles as well. And then we also have adults back to spawning habitats, and sometimes juveniles and late bloomers will follow as well. But we also see other types of movement between habitats, such as from spawning habitats to adult habitats, and movement from nursery habitats back to spawning habitats. And all of those movements, those secondary movements, would be considered contingents. So fish that exhibit contingent behaviors could experience energetic trade-offs that promote future success of their species. Which brings us to the main character in the story, the rainbow smelt. So rainbow smelt are ecologically important forage fish that are known for their iridescent coloration and their distinct smell. Fun fact, they smell like cucumbers when you take them out of the water. But they rarely grow more than 25 centimeters, and they can be recognized by their long, slender bodies, really long, pointed heads, and they also have a superior mouth, which means that their mouth faces upwards. And so although they're really small, they're pretty voracious predators, and they hunt with a mouthful of really sharp conical teeth. And another distinguishing characteristic is the presence of an adipose fin, which can only be seen in 3 families of fish: the salmonids, osmerids like smelt, and ictalurids, which are catfish. So to understand smelt a little bit more, we need to kind of look into their life history. So rainbow smelt are an anadromous species native to North America, and adult smelt will migrate upriver from offshore environments to spawn, and then those eggs will stick to stream bottoms and hatch anywhere from 7 to 21 days, or 1 to 3 weeks, after laying, depending on water temperatures. And then the currents will carry these larvae downstream while the adults return to the bays. And then these larvae will eventually grow into juveniles throughout the summer, and then adults and maturing juveniles will move offshore to cooler water in the fall, and then back up into rivers as the winter progresses to spawn in the spring. So like a lot of fish species, this migration is dependent on seasonal cues like water temperature, light availability, tidal flux. Here in New England, we have really extreme tidal fluxes, so that plays a big role, which we'll talk about later. Smelt reach sexual maturity around 2 years old, but they've been known to complete spawning migrations even earlier than that, and we'll also get into that a little bit more. So smelt contribute to popular recreational ice fisheries all over New England, and at one time smelt were brought in by the barrelful to actually feed families, and then excess was utilized as fertilizer on fields to boost crop harvest. So smelt have been in decline since the 1980s, and they were actually listed as a federal species of concern by NOAA in 2004. So previously, rainbow smelt have been seen as far south as the Chesapeake Bay, which you can see here in the pink on the map. But now you can only see them from the Long Island Sound moving north into the Canadian Maritimes. So NOAA lists the biggest contributors to the decline of rainbow smelt in New England as overharvesting, water quality issues, habitat degradation, inaccessibility to spawning grounds, and even possibly disease. So as we know, New England is famously known for logging and paper industries, which clogged up rivers all over Maine and New Hampshire with dams, culverts, and pollution from all of the pulp mills. So with the introduction of dams and culverts to small waterways, smelt can no longer access previously established spawning grounds. So in other areas where spawning grounds are actually accessible, like here, eggs may be smothered by excess sediment from road construction or even road salts. And so excess nutrient runoff in rainwater also promotes algal growth in shallow streams, which prevents smelt eggs from sticking to rocks, which you can see pictured here on the lower right-hand side. So following NOAA's designation as a species of concern in 2004, New Hampshire Fish and Game actually designated smelt as a special species of concern within the state itself. And so for species like rainbow smelt that are actually experiencing this heavy decline, movement could be the key to their survival. So access to optimal growth conditions and spawning grounds is essential for them to survive, but this also comes with a cost, as we've kind of talked about a little bit. Movement between habitats exposes fish to environmental stressors and makes them more vulnerable to physical barriers when trying to access these essential habitats. So we know that they're moving, but how do we actually track that movement? So a variety of tracking methods exist with different spatial and temporal scales, as you can kind of see here. If we take a look at the broad category of satellite, GPS, and telemetry, we can look at spatial scales of 1,000 kilometers and temporal scales of up to a year. But when we consider a more specific method of tracking, like acoustic telemetry, we shorten our spatial scale to about 10 kilometers with tags having really varying battery life depending on the size. So using telemetry, we face a lot of limitations, such as tag battery life, the locations of receivers, detection range, the receiver array itself. Sometimes we lose receivers, and short temporal scales for detection. So while telemetry gives us really valuable information on seasonal migrations and really fine-scale habitat use, techniques that utilize hard parts of organisms allow us to study movements over entire lifespans. So by utilizing hard part chemistry, we can decipher large-scale movement patterns over really long temporal scales for lifetime habitat reconstructions. So hard parts, or structures like dorsal scales, eye lenses, and otoliths, can reconstruct movement patterns for migratory species like smelt. So here we focus on the use of otoliths to reconstruct migration history, and due to the concentric and seasonal growth of otoliths and the integration of trace elements from the surrounding water into the calcified matrix of the otolith, they're commonly used in age, growth, and chemical history studies to aid in sustainable fisheries management. So I'm talking a lot about otoliths, but what actually are they? So the word otolith comes from the prefix oto, which means ear, and the suffix lithos, which means stone. So essentially, otoliths are ear stones found in the brain cavity of fish that aid in hearing and balance, and they look like that little guy up there. So growth rings form in a similar fashion to rings on a tree, and since each growth increment is a reflection of the surrounding environment at that time, sort of like you're looking at a snapshot, Chemical analyses can be paired with age estimation to recreate a detailed chronological record of a fish's life. So here, if we take a core section of that otolith, that's what that would look like under reflected light under a microscope. And so here we can see a sectioned rainbow smelt otolith, a section that I'm very proud of taking. We're looking at the core region of the otolith indicated by the deep V here of the sulcal groove. And so at the peak of that V is the core or the birth region of the fish. So it starts forming actually when they're in the egg. And then we have these dark opaque banding or summer growth, which is really rapid quick growth, or the lighter, more white bands indicative of winter growth or slow dense growth periods. And like I said, this was imaged under reflected light, so the opposite would be true if you're looking at transmitted. So from otolithic core sections, we can gather elemental data here, and we can use known patterns for these elements to be able to distinguish migration patterns over ontogeny. And so since otoliths are composed of calcium carbonate, it's actually been documented that when strontium and barium are incorporated into the otolith, they will replace calcium in the calcium carbonate matrix of the otolith itself, which makes them really useful as environmental tracers. And for that reason, we'll present calcium element to calcium ratios moving forward. So several studies have investigated the effects of strontium and barium uptake into otoliths, but general patterns that have emerged in the literature is that strontium has a positive relationship with salinity. So this just means that as salinity increases, so does the amount of strontium in the water in the otolith and the water outside, which makes strontium a really excellent marker for marine environments and looking at habitat shifts into marine environments. But barium, on the other hand, has been documented to have the opposite relationship with salinity. So when salinity increases, barium really rapidly decreases. That makes barium a really great marker for freshwater residency. So although these patterns appear really simple and kind of universal, the ratios of strontium to calcium and barium to calcium, they are very, very system specific. So it depends on what body of water that you're working in. And they're both really heavily determined by the amount of dissolved elements, and they're also driven by the bedrock geology of that study system. So elemental ratios and incorporation rates are also driven by factors like temperature, metabolism, diet, which also have to be acknowledged when you're considering chemical structures as tracers. So because of these factors, individual life histories are going to vary, but variation also exists among different species, which only highlights the importance of species-specific life history reconstructions like this. So to put these chemical tracers kind of into context, we have a hypothetical estuary with existing chemical gradients, which we would expect to see the fish exhibiting as they move habitats. So if our fish swims into the marine environment, which has high strontium concentrations, we would expect to see that signature reflected in the otolith, shown here by a red fish. So then, if we travel into the river, we would expect to see high barium, represented by a blue fish. But if we swim into the estuary now, we're expecting intermediate strontium and barium signatures. Some funky stuff happens with elements when we get into estuaries. So our fish would look a little bit more purple, kind of a mixed signature. So with that being said, that brings us to the need for my work specifically, combining stable isotope analysis and otolith chemistry to determine habitat use in rainbow smelt in Great Bay, New Hampshire. So the questions we wanted to answer were, are we able to determine residency and movement patterns from smelt for strontium and barium ratios from core to edge transects using salinity thresholds for each element? It's a lot of words, but I promise I'll explain it. So we also wanted to know whether there was a difference in movement between different life stages and different sexes. And we also wanted to know if stable isotopes were useful in defining habitat use in the species itself. So to set the stage, the study took place in Great Bay, New Hampshire. So Great Bay is a relatively shallow intercoastal estuary that's really well mixed due to really strong tidal influences. So the salinity in the estuary collected from the long-term monitoring station, which is here in that little yellow triangle there, varies on 6-hour time intervals with changing tides and can be anywhere from 0 to 34 from low to high tide, which you can kind of see here, which we're conveniently missing salinity data for our study time. So in a study done by Pearson et al. In PrEP, water samples were taken in triplicate to determine chemical gradients throughout Great Bay. So red circles represent river sampling sites, and they correspond with red over here. And then blue are estuary sites, purple are exit or marine sites. So as you can see from the graph to the left, there's a lot of variability in the river samples in both strontium and barium values, while the estuary and exit samples are very similar to one another with really low variability. So you can see here that this data is really far spread. These two are very similar to one another. And you can also see that here on this graph as well. The river data, we switched colors, so river is now blue, estuary is purple, exit is red. So you can see there's really no variability between the estuary and the exit, but the river is experiencing really high variability in barium and strontium both. So there's a lot of variability in the river samples, like I said, and we'll talk about that a little bit later. So rainbow smelt were collected from 4 tributaries in Great Bay during the New Hampshire Fish and Game annual FikeNet survey and Citizen Science Creel survey throughout the winter in 2021. Fish were collected from FikeNet sites in the Bellamy, the Oyster, Squamscott, and Winnicott rivers, which are marked by the yellow squares on the map, and by Creel survey in the Oyster and Squamscott rivers, marked there by the purple circles. So fish were either sampled as full bodies or just heads, depending on the method of capture. So Creel surveys, we let the fishermen keep the bodies, so we just get the heads for the otoliths. And so once the fish were collected, morphometrics were taken, and fish were transported to UNH for otolith removal, and then the bodies and otoliths were transported to UNE for further analysis. So otoliths were embedded in epoxy and thin sectioned into approximately 1 millimeter cross sections with a low-speed isomet saw. And then after sectioning, otoliths were polished to expose the core. And once they're polished, they look a little something like that. And then they're mounted onto glass slides, and then digital images are taken and collected for elemental analysis at the Jackson School of Geosciences at UT Austin. So we used laser ablation inductively coupled plasma mass spectrometry, or LAICPMS, to get chemical concentrations along a core-to-edge transect of each otolith. So the circles here that you see at the bottom of the screen are our standards that we run in between samples to sort of kind of reset the laser in a way to ensure maximum accuracy across our transects. So this is what the drawer looks like when it goes into the laser chamber. It stays like that throughout the whole analysis. And then our otolith is ablated from core to edge by a small, powerful laser, and once run through this big machine, we get elemental concentrations out of that. So we also tested the accuracy of doing a single line transect and how representative that actually is of the whole life. So we completed 2D maps using the same technique over the course of the otolith. So instead of just doing one line from core to edge, we actually covered the entire otolith back and forth. And so we used the largest male and the largest female in the study to see if there were any patterns between sexes. And so as you can see from the maps, strontium and barium appear in oscillating patterns all the way around the entire otolith, which just confirms the accuracy of a single line whole life transect. So we're getting the same thing doing this as we are doing this. So the raw data are then smoothed to remove any high frequency noise, and the edges are trimmed to determine the true edge of the otolith based on calcium and indium values. So once our values are trimmed and converted to a molar ratio, our transect looks something like this, where the red line is strontium values along the transect and the blue line are barium values, but we can't really get much out of this graph yet. So in order to get what we want out of the data, we have to do some calculations. So we use partition coefficients to compare otolith elemental concentrations to water concentrations in the environment. And our partition coefficient is just the fraction of elements from the water that are incorporated into the otolith's matrix as a fish grows. So the first equation here that you see on the screen, we can see that a partition coefficient value, or a D value, is calculated by the concentration of elements in the otolith divided by the concentration of elements in the surrounding water at the time. So a really unique part of this study is that we have water values taken for the specific system when the fish were collected, which is really special because it makes our study very species and system specific, like I was saying earlier. So we did this for both strontium and barium, and once that was done, we needed to figure out what our threshold values in the otolith would be in order to actually determine where these fish were. So by establishing a threshold for each element, we can tell what elemental concentration defines freshwater or saltwater residency in our specific system. So by rearranging the partition coefficient equation, like you see down here, we can back calculate a threshold value above which will elucidate freshwater or saltwater residency. So to know a little bit more about life history, we also had to age our fish. So to age the smelt samples, I talked to a Rainbow Smelt age and growth scientist with the Mass Department of Marine Fisheries, and we aged our smelt using 1 dark ring and 1 light ring to equate to 1 whole year of growth. So here we can see the first year of life, and then on this fish, we can see the second year of life. And we also used age to determine residency in days in each body of water. And so for the sake of time and for the sake of everyone's brains, I will not go through the calculations, but if anyone has questions about it, I'm happy to talk about it after. So since most of our fish were in the age 2 year class, we decided to further split the analysis into the first year of life and the second year of life, determining habitat use. So this is what our transect looks like now that we have our thresholds and the marker where the first annuli was identified. So remember that first year of life here. If this is a core to edge, this is where that first year of life is going to be happening. So for strontium, above the threshold delineates marine residency, and for barium, above the threshold delineates freshwater residency. And then estuarine residency was assumed when both transects were below their respective thresholds. So we used a hierarchical cluster analysis to determine residency in the first and second years of life. And so seen here are the metrics we use to plug into our cluster for each year. So we use the number of crosses, seen here as the number of times each transect crosses that threshold. So separate for strontium and barium, but every single time they cross this line. And then we also use the sum of values, which is just simply the sum of all of these values across each transect. And then we also use the duration over the threshold, which is any of these peaks and how many days this number of microns would equate to, first time spent in that body of water. So now we actually get to address the first question. Can we determine movement patterns from these salinity thresholds that we calculated? So using the equations we talked about earlier, I determined a D value, or partition coefficient, for each river and averaged those values to get an overall D value for each element representative of the entire system. So using these D values, I back calculated 3 thresholds to explore the variation between each, but as we move forward in the analysis, we'll use kind of that median threshold there because that makes the most biological sense rather than, high and low. So these threshold values can be seen here are a prime example of a migratory fish. So we have the thresholds marked here. We have 3.31 for strontium and 1.29 for barium. And so we can see a really clear movement here into the marine environment and a very clear shift into a freshwater environment directly after. So all of these metrics seem kind of simple. We're just kind of talking about it in general. But if you look here at the edge of the transect, we can see that both of those lines are above both thresholds at the same time. So anomalies like this in the transects are what make otolith chemistry more interesting and definitely more complicated. So as we saw on the previous slide, now that we have the ability to assign habitat use to each fish's profile, we can use the same technique to investigate what smelt are doing in the first year of their lives. So movement within the first year of life is currently unknown since tag data provides movements for adult smelt throughout the estuary. So we know what we would expect juveniles to do, right? We would expect them to hatch, float down into the estuary, live there until they're ready to move offshore and then upriver to spawn. But what do we actually see them doing? So we can see from my really super colorful graphs here that when we line up the distance of the first annuli at 0, so every single fish in the study for females and males is lined up with their first year of life right at 0 here, we can clearly see a pattern, right? So we see that most of the females appear to be making that offshore migration right at their first year of life as they're laying down that first annuli. And you can see that kind of opposite pattern reflected in barium here. So females make a lot of sense, right? But when we look at males, they're kind of all over the place. A lot of variation, a lot of spread across the data in both graphs. So they have a lot more variability, but how do we actually quantify any of this? So the metrics from the previous slide, like I said, were plugged into 2 separate hierarchical cluster analyses to see what kind of separation we would get if we consider strontium and barium and those metrics as different factors. So when we run those metrics through a cluster analysis in R, this is how our data look. So for year 1, the cluster identified 3 distinct groups, which you can see here by the different rugs, so blue, purple, and red. So looking at our strontium data here, we can see that differences in all the groups across all the metrics, but the only significance lies in this group up here for strontium. But when we look at barium, we would kind of expect to see this stepwise pattern that we see here mirrored, right? So we would expect it to be high, medium, low here instead of high, because we would expect strontium and barium to have inverse relationships to one another. But as you can see, we don't actually see that. So it could be due to a number of things, but we'll discuss that a little bit later. But looking at our metrics, we can hypothesize that there are 3 separate contingents that kind of appear within this population that utilize different habitats in early life. So are there any actual obvious advantages to smelt contingent behavior? So when we look at advantages for different migratory strategies, growth is a really major component to gaining an advantage super early in life, right? So the bigger you are, the quicker you're able to become not eaten. So simple as that, right? The bigger you are, the less likely you are to get eaten. So here we can see that even though venturing into the marine environment as a juvenile presents a lot of risks to small fish, there's clearly some sort of benefit happening because we see these fish experiencing significantly higher otolith deposition, which is directly related to total length, during that first year of life. So we can see here that this third group here is experiencing significantly higher growth than these 2. So there's clearly some sort of benefit going on. But when we look at males and females, although we've seen difference in adult smelt movements between sexes in telemetry studies, we don't see a whole lot of diversity between sexes within the first year of life when either element is considered. And we kind of expected this, right? They're not sex segregated when they're this little, they're just kind of trying to survive. So when we look at the second year of life, we have the same metrics filtering into a cluster, but now the cluster identifies 2 different groups. So we have this first group here in the light blue, and then the second group here in the dark blue. And so looking at just strontium at the top, we can see that group 1 displays significantly lower strontium than group 2 in 2 out of the 3 metrics, but there's a lot of variation across all data. So if you look at these bars here, you can see that they kind of encompass each other with variation. But when we look at barium, we see that it's again mirroring strontium in a way that we really wouldn't typically expect, and again, the variation is really, really wide across each of these. So when it comes to growth advantages for these 2 groups, group 2 experienced significantly higher growth than group 1, but we can see that the variation in group 1 encompasses the entirety of group 2, which just means that there's a huge spread across the data in each group. And again, when we look at males and females, we see no difference between habitat use between either of them. So for the first question, we were able to determine partition coefficients and establish salinity thresholds above which we were able to determine residency. So here we can see that this signature is indicative of a movement into the marine environment, directly followed by a shift into a freshwater habitat, which is likely a coastal migration in the red, followed by a spawning run in the blue. So like all approaches, there are some caveats to consider when we're using calculated values to interpret habitat shifts from otolith chemistry. So, although there's sufficient evidence in the literature displaying a positive relationship between strontium and salinity and the opposite for barium, these values are really largely based on the complexity of incorporation rates, temperature, and the amount of dissolved elements in the water itself. And there's also been documentation that strontium and barium values plateau around 20 in most systems, making them useful at really low salinities, but the relationship becomes really complex at kind of those in-between salinities. So partition coefficients are also species and system specific, but they're calculated for the purpose of estimation, and the back calculation of thresholds is really just kind of an arbitrary number that we would expect these values to represent that external environment at that time. So the reality is that there are a number of external and internal physiological processes that could affect the integrity of these estimations and therefore kind of the interpretation, but they're just kind of caveats, so that doesn't mean that it doesn't make sense, right? So for our second question, we defined 3 contingents within the first year of life. So these contingents could provide spatial separation within the entirety of the population, where individuals possess exploratory behaviors that ultimately increase their overall fitness. So the contingent hypothesis has been evaluated in other species like striped bass and southern flounder, but it's yet to be fully evaluated in rainbow smelt themselves. So our data points to the presence of contingents in early life stages, whereas, so we can see that here, 3 separate contingents, right? We have these 3 different groupings that we see in these different signatures. But when we move into the second year of life, we can kind of see that it appears to see some schooling behavior that becomes present as these fish kind of approach sexual maturity and start to really school together. So it's been noted in previous studies by Enterline et al. 2012 and Chase et al. 2019 that smelt could reach sexual maturity early and actually begin these spawning runs before they're sexually mature or before that first year of life. So there isn't a ton of information in the literature about this yet, so we're still kind of looking into it, but it could be a possible reason why we're seeing this. So when we look at growth in the first year of life, our fish that ventured into the marine environment with those high strontium signatures have significantly higher growth rates than those other 2 groups, which seems like something that would kind of go against the norm because you're kind of you're taking the risk moving into this environment. It doesn't really make sense that they would actually survive, but the ones that do grow to be a lot bigger. But it's actually been documented before by Sarah and Dodson in various publications that smelt in the St. Lawrence River in Canada actually prefer to exist in this kind of marine environment in this estuarine turbidity maxima, or the ETM. So this is an area of extreme turbidity that provides juvenile fish with increased protection from predators and an abundance of food particles like suspended matter and plankton. So although there isn't current evidence of an ETM in Great Bay, there's been several studies that thoroughly document high amounts of suspended matter that actually peak in the spring when smelt are hatching and beginning to kind of drift into that zone in the estuary. So the separation between year classes could be driven by the metrics that we plugged into our own cluster. The metrics could be skewed by large spikes in strontium or barium, which you kind of see here, these really large kind of peaks, which in turn affects the interpretation of values for each element. So if we were to redo this, which we did because we're writing a manuscript, we would use a measure of central tendency like the median instead of the sum to kind of get rid of that skew there. But I also want to bring your attention back to the phenomena when both transects are above the threshold at each time. And this could have a variety of explanations, but the strongest reasoning behind this is likely that the increase of barium uptake in a higher salinity environment like an estuary. So when it's common belief that in otolith chemistry, that strontium will outcompete barium for binding sites in fish otoliths. But a study done by DeVries et al. In 2005 displays that elemental incorporation of barium in the otoliths of black bream may actually be increased due to defects in the otolith structure caused by ambient strontium in the water. So barium is a much bigger element than strontium. So it's the equivalent of trying to fit 2 puzzle pieces together that don't quite belong. But when we bring ambient strontium into the equation, it causes defects in the otolith matrix, which allows barium to bind much more easily than strontium. But this could be true, or it could also be due to the fact that barium is notoriously misbehaved in estuarine environments due to the amount of suspended particles and riverine sediments that are supplied to the watershed itself. So historically, sex comparisons aren't commonplace in otolith chemistry, which makes our study really unique, not only for sex comparisons in Rainbow Smelt, but kind of in otolith chemistry as a broad study. Although we noted no difference in habitat use between males and females, based on our otolith chemistry, that doesn't necessarily mean that the fish aren't utilizing different habitats at different rates. It just means that elemental incorporation into the otolith wasn't able to keep up, so to speak. So with rapid movements and changes in habitat during spawning seasons, these elements just can't really track those quick movements. So telemetry data from Pearson et al. In review has documented that males, on average, spend about 17 days in rivers during spawning season, which is almost 2 times longer than females. And 2 males in that study were also seen visiting 3 different spawning rivers, and they were actually traveling together. So on this figure here to the right, I just want you to notice the movement between spawning rivers, indicated by the arrows here. I know they might be hard to see, but they're little points. So that indicates the direction of the flow. So from the Squamscott to the lamprey, there's movement. And it's also, these are the relative number of detections for each. So you can see that most of the movement is going out into the estuary or from the estuary to the exit. Kind of this flow of events here. But we can see movement in between each individual river as well. So from this, we can conclude that otolith chemistry is definitely more useful for longer-term studies and habitat use over ontogeny, whereas telemetry has proven really useful for rapid seasonal movements that we can't pick up with otolith chemistry. So if we kind of switch gears to another method that we use to determine habitat use, we can look at stable isotope analysis. So isotopes like carbon-13 and nitrogen-15 from white muscle tissue can offer information into what an organism was consuming and can help estimate trophic positioning. So carbon-13 is typically an indicator of primary producers in a given habitat. So we would expect a typical freshwater environment to show a different carbon-13 signature than a marine environment and vice versa, hence these different end members here. So nitrogen-15 is typically an indicator of trophic positioning. So in the case of our smelt, we would expect it to be pretty low since we aren't looking at a really high-level predator. We're looking at more of a forage fish for the area. So like otoliths, muscle tissue reflects chemical gradients and changes in the environment, but the main difference between these 2 methods is that otolith chemistry can be used for life history reconstruction, so really long periods of time. But white muscle tissue may only show the last few months to a year of a fish's life due to the equilibration timing of those chemicals in the tissue. So equilibration is very species dependent, but for a small species like smelt, we're probably looking at about a year for the tissues to fully reflect the environment that they're in. So essentially, if we have 2 end members, freshwater and saltwater here in the blue and in the red, we can assume with appropriate equilibration, we should be able to determine where the fish were within that sort of time frame. So without appropriate equilibration timing, our fish will be in kind of a transitory state, which is here in purple. So if we're staying in the saltwater environment for a while, we would expect them to look red. Same with the freshwater, we would expect them to look blue. But if we're transitioning habitats, or if we're, kind of a newcomer to a habitat and our muscle hasn't turned over completely, we're going to have those sort of mixed signatures. So provided the tissues have reached equilibrium within the environment and an isotopic gradient actually exists, we can extrapolate recent habitat use and connectivity from carbon-13 and nitrogen-15 values in soft tissues as well. So using stable isotope analysis and otolith chemistry together, we can provide a wealth of information about habitat use and connectivity between fish populations. So to prepare the isotope samples, I excised a chunk of dorsal white muscle tissue from each of the 48 fish that we had whole bodies for. And then the setup looks something like this. Once the muscle tissue was taken out, the sample was scraped clean of skin and scales because they're going to show different chemical signatures than muscle will, and freeze-dried for 48 hours. And after freeze-drying, samples were ground into a fine powder and packaged in tin capsules and sent off to UC Davis for analysis. So when we got this data back after a very long time waiting, we investigated carbon-13 and nitrogen-15 signatures of fish to see if we could determine capture location from these signatures. Because remember, we're looking at more recent life now with these. So, however, you can see in this graph, all the rivers were really pretty evenly mixed. There's really no different chemical separation between each of the rivers. So to double check on this, we also repeated this process with the last 25 microns of the otolith edge, which represents the same time frame as what we would be looking at in the tissues. And we were again unable to see any chemical differences between the rivers. So in tandem, we couldn't use the chemical signatures to distinguish rivers of capture. We also looked at the split between males and females in stable isotopes, and again, we really see no separation based on these isotopic values as well. But what we do see from stable isotope analysis is a significant correlation between nitrogen-15 and total length. So as a fish grows, its trophic position increases because the prey that it's eating is changing. So the bigger the fish, the higher the nitrogen values will be. So from these data, we can confirm that nitrogen-15 was useful for determining trophic position of rainbow smelt, despite its lack of success in determining habitat use. So the point highlighted in red up there is considered an outlier. So we've done a lot of analysis on this, and if we remove that outlier, it shifts the relationship ever so slightly and becomes significant. Whereas with the outlier, it was non-significant, and with it, it is significant. So what does all of this mean in the context of Great Bay? So Great Bay is a really dynamic, well-mixed estuary that flushes on 6-hour tidal cycles. And so it's really likely that our isotopic analysis didn't show any separation between habitats due to mixing. So with flow rates of up to 2 meters per second, which is really fast if you know anything about estuarine dynamics, and tidal ranges of up to 11 feet, the probability of the presence of an isotopic gradient throughout Great Bay is actually really low. So therefore, all our fish appear to have mixed signatures, aka they all look purple. So on top of that, if smelt do indeed mirror the duration of a year for muscle tissue turnover, it would be impossible to discern fine-scale movements from stable isotopes because there's no way that they would be staying there in that time frame for a full year. But the positive correlation between nitrogen and total length is really promising in the isotope data because it shows us exactly what we would expect. We would expect nitrogen to increase as fish get larger because as juvenile smelt go from eating low on the food chain, phytoplankton, zooplankton, copepods, to eating and cannibalizing juvenile smelt, other small fishes and crustaceans, nitrogen is established in the literature to document trophic shifts in a variety of species, from small invertebrates to really large pelagic predators like tuna. But what does all of this mean in a much broader context? So to summarize our trace elements results in a broader context, we may have seen varying patterns between strontium and barium due to abiotic factors like temperature and diet. Miller 2011 provides evidence that strontium values are higher in freshwater prey items, meaning that about 30% of the strontium incorporated into the otolith in freshwater environments actually comes from dietary contributions and not the surrounding environment. So this same study and several others across the literature also documented effects of temperature and salinity on strontium to calcium ratios and ambient strontium concentrations, but they've documented little to no effect of these variables on barium to calcium ratios or barium in general. But confounding evidence exists in the literature about the effects of temperature and diet on otolith chemistry, so it's definitely an area that needs to be explored further in a species-specific context. And similar to stable isotope equilibration times, otolith trace elements have been documented to take about 20 days to reach equilibrium with the surrounding environments, and again, variation is found between strontium and barium in the environment itself. So to summarize our stable isotopes in a broader context, our study and several others display evidence for difficulties in using stable isotope analysis in really small tidally dominant estuaries. But while we provide no evidence of an isoscape in a tidally dominant estuary, that doesn't conclude that there isn't one there. Further study is needed for Great Bay and other well-mixed estuaries to determine the likelihood of isotopic gradients in these estuaries. Tissue turnover rates are essential to understanding the complexities of metabolic processes, and species-specific turnover is needed to complete habitat reconstruction. There's no current tissue turnover rate established for smelt, so that's a huge issue that we need to kind of work more towards. But also, we must consider the idea of variation between life stages. Isotopic turnover has been documented to vary between larval, juvenile, and adult phases, with growth being the major driving factor in isotopic turnover in those young, early life stages. So to kind of wrap all this up, by utilizing otolith chemical signatures and continuing to validate this technique across systems, we can continue to enhance our ability to understand interpopulation differences. And the appearance of contingents within a population is becoming more and more recognized, and otolith chemistry has been a really great tool in identifying contingents within migratory populations. So contingents allow fish to respond to these environmental changes, and they may allow plasticity in migration timing and movement between different habitats. So not only do adult fish need access to spawning and feeding grounds, but this is kind of looking into juveniles and young of year fish actually needing habitat connectivity as well, because they've been observed to migrate significant distances across these critical habitats. And studies such as this contribute to the idea that it is essential to understand the movement requirements between life stages to inform management strategies that encompass the entirety of ontogeny, not just adult life stages. So acknowledgements again, I just kind of want to thank everybody, especially Ocean Studies, for having me come today and do this talk again. My funding source, Sea Grant New Hampshire, all of my really great collaborators, faculty, staff, family, everybody. And with that, I'm happy to take any questions.
I have a feeling that they're at the door, but we take one question maybe, and then Lex, would you be willing to stand around and maybe outside and
for
chat
sure.
with people afterwards? Okay, so Allie.
Hi, I'm Allie. I'm a second-year marine bio major. Were there any ethical concerns with the study, given that for a high concern endangered species, you had a relatively large sample size? And also, what was done with the whole body specimens you collected once you extracted the otolith?
Yeah, that's a good question. The ethical concerns, all of the permitting is through New Hampshire Fish and Game, so they actually do everything with NOAA. They monitor the population. They can only collect a certain amount. They only collected 79 over the entire year, which is really, really small. Usually their sample size is like over 200. They're collecting from all the different rivers, and they all have returning spawning runs. So all of it is permitted and everything is managed, so they're only allowed to take a certain amount. What was the other part of it? What was done with the whole body specimens once you extracted the otolith? I have them. They're all in frozen little baggies in my lab freezer currently. So the otoliths were taken, and then the bodies were sent to me for me to take muscle tissue out of them. But my advisor actually wants to do some eye lens analysis on them if it's possible. So he's trying to do more stuff, and he's really big on utilizing like the entire body of things and not putting anything to waste. So.
Okay, so I think we're going to have to stop so we can transition to the next group. But let's give Lex a big round of applause. And then we can just head out to over by the spruce room if anyone has.